All righty. Good morning, everyone, and welcome to session 2 for today. Thank you for joining us. If you're just joining us for your first session of today, I'm Johnny Scott with Syngenta. I'm an MABA board member for the past 5 years and the secretary of the board. I'll be your moderator this morning for session 2 as well. Thanks again for participating in the MABA Great Montana Ag Rally. We hope that you found these informative, and we will continue these throughout the winter and spring. First off, a huge thank you to BASF for sponsoring this session. We can't do it without our sponsors, so thank you, BASF. So first off, let's start with a message from our sponsor, BASF.
Hello and welcome to this session from BASF. My name is Paul Clemmy. I'm the product manager for inoculants and biologicals with BASF Seed Treatment. I grew up on a farm in central Iowa where my family still farms, and I've been in the agriculture business my whole career. The last 12 years, I've been in the inoculant industry in sales, marketing, business development, and product management. I joined BASF because of some of the great new innovation that we're bringing to market, especially in the inoculant space. What I'd like to do for the next few minutes during this session is talk to you about one of those new innovations, Nodulator Duo Solid Core Granule Inoculant for peas and lentils. One of the things that we'd like to provide as a gift to you is this tumbler for attending this session, as well as learning more about Nodulator Duo Solid Core Granule Inoculant. So all you have to do is email your name, address, city, state, and zip to the Montana Ag Business Association email here. While you're doing that, I'll play one of the ads that we have running on your local radio stations talking about one of the great benefits that Nodulator Duo brings your growers.
Yeah, so I use new Nodulator Duo Solid Core Granular Inoculant on my peas and lentils.
This guy, he's full of seeds.
3 more bushels per acre.
Ha! You're full of seeds, pal.
Well, yeah, my crops had better nutrient uptake and a root strengthening biofilm.
You're full of seeds.
Right, I know. I had bigger and fuller pods.
You really are full of seeds. Full of seeds.
Try Nodulator Duo SCG by BASF for bigger, better yields. BASF, we create chemistry. Always read and follow label directions.
Hopefully you enjoyed that ad and look forward to hearing that on your radio stations. As I mentioned, Nodulator Duo is brand new for 2021. It has been available in Canada for the last 3 years, and we're excited to bring it to the US. Nodulator Duo has 3 unique features for your pulse growers. This technologically advanced solid core granule that I'll talk about a little more in the next slide, the high-performing rhizobia strain specific for peas and lentils, and then thirdly, this new plant growth promoting rhizobacteria, Bacillus subtilis, that brings some great nutrient uptake as well as plant growth development that you can get in Nodulator Duo. So the granule, solid core granule, is the first thing that I'll talk about. Here on the slide, you can see we have a clay-based hard granule surrounded by layers of the rhizobia, the bacteria, as well as some polymers and overcoat protection to allow that rhizobia to survive and then thrive once it does get into the soil. It is engineered to deliver the product through your air carts, through your seeders. It's durable, uniform in size. And you can see here in this picture, the peat products that a lot of competitive products may be utilizing as a base sometimes can have some dust or have some flowability issues. With solid core granule, you should not have those issues and can have some great improvement when it's flowing through your customer's equipment. The second key thing with Nodulator Duo is this rhizobia strain. It's specific for peas and lentils. It was developed in Canada, and we see increased nodulation, increased nitrogen fixation. This picture here on the bottom shows a competitive inoculant versus the BASF strain where you've got nodules throughout the root structure as well as a larger root mass. The third and the duo part of Nodulator Duo is this new bacillus. This bacillus provides or forms a biofilm on the root. Here you can see a water control on a petri dish versus the right-hand side where the biofilm is. What this biofilm does is it allows the plant to intake or uptake water and nutrients more readily. It helps mitigate abiotic stress, and it improves that relationship between the rhizobia, the bacteria for nodulation, nitrogen fixation, and the plant. The result is you get higher yields. When you look at this plant growth, here's a slide that talks about some of that enhanced plant growth. The blue bar is Nodulator Duo Solid Core Granule, and the dark bars are competitive products or the old formulation of Nodulator. Early season results, mid-season results, significant differences in plant growth above ground as well as below ground, as you can see in the picture on the right-hand side. Of course, yields is what pays the bills. And as the radio ad talked about, we see about a 2 to 3 bushel yield response with Nodulator Duo Solid Core Granule. Nodulator being in the blue, competitive products, whether it be a base, low rhizobia, or what's considered somewhat of a premium product, we see a significant yield response with Nodulator Duo. Again, more nodules, greater root structure, all adds up to more yield. Next, what I'd like to do is play a short video that talks about or shows the biofilm as I talk about it. Again, this root strengthening biofilm really improves nodulation as well as the ability of key plants to be able to take up nutrients and expand their ability in the soil to utilize the water as well as nutrients that exist in the soil. You can nodulation throughout the biofilm, expanding the reach of those roots. The next couple slides, I'll talk about our portfolio and some inoculant handling best practices. We have a complete portfolio for pulses as well as chickpeas. Nodulator Duo Solid Core Granule, as I just talked about. We've got a couple package sizes, 850-pound tote bag. Granule, the old formulation of Nodulator, liquid as well as peat. For chickpeas, we have a dedicated strain for the chickpea clay granule. So if you've got growers that are utilizing chickpea, make sure that you've got the right granule for them. Again, same package sizes as the Nodulator Duo for peas and lentils. When it comes to grower efficiency, the Nodulator Duo brings some great advantages here for your growers. If you're on a 7-inch row spacing, 4.7 pound application rate, a grower can cover 170 acres per that tote. If they're on a 10-inch spacing, 245 acres at 3.3 pounds, which is significantly less than some of our competitors that are in that 5 to 6 pound range. The last couple slides talk about the importance of handling inoculants properly compared to chemical products. They are living organisms, and so you just need to handle them a little bit differently. Proper storage and handling are key to make sure that you get the performance that you want. BASF does have the highest standards for all of our inoculants and recommend that you do handle them as per recommendations. And one of those recommendations is to make sure that the products don't freeze. The granule formulation is the hardiest and can handle exposure to hot and cold conditions, but it's important to not let it freeze. We do recommend that you store it in dry conditions with good air circulation and only stack the pallets one high. The last slide I'll talk about is the BASF's commitment to quality. We've got quality products. We've significantly improved our manufacturing capabilities to deliver products where and when they're needed. We've got the ability to meet market demand, and we're always looking to adapt, invest, and improve our biological products. Nodulator Dual Solid Core Granule is one of those new innovations that we hope that you'll like. And with that, I encourage you again to send an email to the Montana Ag Business Association to receive your free tumbler. Listen for the radio ad that's playing, and also encourage you to reach out to your BASF representative to learn more about Nodulator Duo. Enjoy the rest of the sessions, and we look forward to working with you throughout the next few years, bringing innovation to the inoculant space.
I had bigger and fuller pots.
You really are full of seeds. Full of seeds.
Try Nodulator Duo SCG by BASF. Bigger, better yields. BASF, we create chemistry. Always read and follow label directions.
Enjoy the rest of your sessions. Thanks again for attending.
Well, thank you again to our sponsor, BASF, of this session. Now we'll move into the actual presentation with Dr. Uta McKelvy. Dr. Uta McKelvy is the Associate Extension Specialist for Plant Pathology and Assistant Research Professor in the Department of Plant Sciences and Plant Pathology at Montana State University in Bozeman. Uta obtained her PhD in plant pathology from the Montana State University in 2020, where she worked on dynamics of the wheat streak mosaic disease complex in the agricultural landscape. In her role as extension specialist, Uta focuses on diseases of field crops, such as small grains, pulses, fungicide use, and fungicide resistance prevention, as well as integrated pest management. Her current research focuses on developing prediction tools for foliar diseases of wheat in collaboration with the National Predictive Modeling Tool Initiative. So with that, thank you again, Dr. McKelvy, for being with us. I will turn it over to you.
Yeah, thank you for the kind introduction and good morning, everyone. Welcome to this session. I'm excited to be here and I'm going to start my screen sharing here in just about a second. As mentioned in the introduction, my current research is focused on foliar diseases of cereal crops, but since that has just started and is virtually in its infant shoes, I'm decided to talk about pulse crop diseases today to you. And so my focus is overall on both persistent diseases that we've known and are familiar with over the last couple of years, but I also want to mention a couple of emerging pathogens that may present disease challenges to pulse growers in the future. I'm starting the slide here just with some information about myself, which will occur later. I highly encourage you to reach out to me with questions that you may have following up on this presentation or any other concerns you may have related to plant pathology. And I've also included some resources that you may find helpful when it comes to disease diagnosis, seed testing, or information on other disease issues. So growing pulse crops can be very profitable, but is also challenged by a whole host of pathogens that cause diseases. Just to name a few, I've listed some here on the slide. Obviously, I will not have enough time to talk about all of those diseases today, so I'm just going to focus on a few that are, as I mentioned before, persistent, occurring every year, or are particularly threatening to pulse crop production in Montana. And the way I decided on which pathogens to focus on, I can disclose that is mostly through my work at the diagnostic lab, the Schutter Diagnostic Lab. We've just written our annual report, which you can find on the Diagnostic Lab's website. And I decided to focus on those pathogens that were coming quite frequently through our lab. I want to introduce you very quickly to the team here at Montana State University that is working very hard to provide new insight and information and management recommendations for diseases, not only for pulse crops, but also for other crops grown in Montana. I'm sure you're very familiar with Mary Burrows, the queen of plant pathology here at MSU, and she has an amazing team of students, lab assistants, diagnosticians, et cetera, that are all working very hard on various subjects. And some of those entities I will talk about a little bit more in the upcoming slides. So the first issue in pulse crops that is quite persistent that I would like to address today is the root rot complex of pulse crops. So this slide here shows what exactly that complex is composed of. So we have different agents that can cause root rot diseases in pulses, some of which belong in the family of the Oomycetes, which are also known as slime molds. One that might be familiar to you by name is Aphanomyces Root, but I'm sure that Pythium and Phytophthora are also not unfamiliar names to you. There are several true fungal pathogens that can also cause root rot issues in pulses, namely Fusarium, but also Rhizoctonia, Alternaria, et cetera, et cetera. And last but not least, we have a couple of nematodes that can cause substantial issues to the root system of pulse crops. Unfortunately, I am not by any means an expert on nematodes, and so I will casually omit this specific pathogen group in today's presentation. So generally, the root rot symptoms, if you're having a bird's view on a field, you may observe them first in parts of the field that are frequently flooded, have a high soil moisture that persists for a long period in the season. And root rots also often occur in areas where soil is compacted, which is quite frequently and also low spots in the field. Over several years, the symptoms of those root rots may expand beyond those specific areas of compaction and high soil moisture. And this expansion and increase in disease severity is often correlated with the number of years that pulse crops have been grown in your field. Here in this picture, what you can see is a field that has been planted to peas, both of those sites. But here on the left, you have a field planted to pea where pulses have not been grown before. Whereas on the right, you can see a field pea, a pea field that has been planted to pulse crops prior. And so these previous years of tight pulse crop rotations have increased the inoculum of these root rot pathogens and therefore exacerbated the symptoms that we now observe in this field. On the boots on the ground perspective on your pulse crops in the field, you will often observe poor emergence, poorly growing plants that are rather short and yellow in appearance. All of these are first indicators that you might be looking at a root rot issue in your field. But to really figure out if that is the causing issue, you will have to dig up some plants and look at the root system, since this is, as the name implies, where the pathogen attacks and the disease expresses. So often the root system is very reduced, having only the taproot and maybe a few lateral roots. The root system is often also discolored in various shades of brown. You will see poor nodulation or no nodulation at all. I have included here a link to a video that you can find on YouTube, which was produced last year by the Diagnostic Lab and MSU Extension, that talks about diagnosis, identification of several root rot pathogens, and also methods to mitigate root rot issues in your field. Highly informative and very aesthetically pleasing. I can recommend you to watch this video. So these symptoms that I described in the previous slides are rather general symptoms for root rots. Now I want to talk specifically about two causal agents of root rots, the first of which being Aphanomyces root rot. Aphanomyces euteiches is the pathogen that causes Aphanomyces root rot. As I mentioned before, Aphanomyces is an oomycete, which is a fungal-like pathogen, which is commonly known as water moles. Aphanomyces root rot and the pathogen itself prefers wet, waterlogged soils. The spores of Aphanomyces, they're called oospores, and they can last in the soil for up to 20 years, making this particular pathogen and this disease very difficult to manage. The reason why Aphanomyces prefers wet and waterlogged soils too is because it produces a second type of spores, which are called zoospores or swimming spores, which literally swim in the water. And so if we have areas in the field where we have high soil moisture, this is where these zoospores can swim from root to root, spreading the spreading the disease, excuse me. Generally, Aphanomyces root rot is favored by wet conditions and cool conditions. The optimal temperature for infection and disease development is around 25 degrees Celsius. Aphanomyces root rot was first detected in North Dakota in 2014, and then 2 years later in 2016 in Montana as well. This first identification in Montana has launched a research project conducted by a graduate student of Dr. Mary Burrows, who has conducted since extensive field surveys, trying to locate fields with Aphanomyces pathogen in them, and also conducting research to find out suitable management strategies to mitigate this disease. Aphanomyces root rot generally is more severe in low-lying areas in the field where we have an accumulation of soil moisture. And as I mentioned before, the more often pulse crops were grown in the recent past, or I mean not so recent since the pathogen can persist in the soil for up to 20 years. So the more often pulse crops are grown, the higher the risk or the higher the inoculum of this particular pathogen in the soil, and therefore the occurring disease. So what is very characteristic for Aphanomyces root rot specifically is the caramel brown or honey brown discoloration of the root system, which you can see here in comparison to healthy roots, which have a rather light pale, light brown or yellow color. As I mentioned before, nodulation is often reduced. Nodules appear pale in color or have not occurred at all. Something that is important to keep in mind about Aphanomyces root rot is that both peas and lentils are very susceptible to Aphanomyces, whereas chickpea is rather resistant to this pathogen, as well as faba beans. So these two pulse crops or these two crops might be a good alternative to incorporate in your rotation if Aphanomyces is a problem in your field. A telltale or a symptom of Aphanomyces root rot that you may observe without digging up the roots from the field is that often the lower leaves appear yellow and pale. The plants are stunted and wilt and die prematurely compared to the healthy plants in the field. So what are some control options that can be undertaken to mitigate the impact of Aphanomyces root rot on your pulse crop production? So the most important thing that you can do is crop rotation. You should do that generally, but especially if you know that you have Aphanomyces in your field, you want to make sure that you have a rotation where you rotate out of particularly peas and lentils, but in general out of pulse crops for several years. Now you have to weigh the options between what is ideal and what is feasible for you. Generally, it is recommended a rotation out of pulse crops for at least 6 years or better 8. But any year or any season that you can include in your rotation where you're not growing pulses is gonna be reducing the disease pressure for Aphanomyces root rot. As I mentioned before, faba beans, soybeans, and chickpeas are somewhat ideal pulse crop options if you do have Aphanomyces but do not want to miss out on growing these legumes. However, specifically with chickpeas, low levels of Aphanomyces root rot are still possible because they're not entirely resistant. Something to keep in mind with mitigating Aphanomyces disease is also that not only crops, but also certain weeds can be hosts to Aphanomyces, thereby increasing the inoculum or at least helping this pathogen to persist in the soil. So you want to make sure to control weeds in your field very closely. Specifically, volunteer weed, white clover, shepherd's purse, chickweed, and field violet are such weeds that can be a suitable host for Aphanomyces. One strategy that has proven effective in protecting your pulse crop from Aphanomyces root rot is seed treatment with ethoboxam. And so this is generally recommended specifically if you know that you have an oomycete issue in your field. Ethoboxam specifically controls oomycete pathogens such as Aphanomyces, but also Pythium, for example. But it doesn't control, for example, Fusarium or Rhizoctonia or similar root pathogens or seedling pathogens. So you want to make sure that you combine this specific mode of action with another one that is effective against Fusarium and Rhizoctonia, etc. Another thing to keep in mind about seed treatments is that they typically are active for only about 3 weeks. So seed treatments really only provide an protection against an early infection of your seed or seedling, but they do not provide protection throughout the entire growing season, which is why it is important to implement more management strategies than just a seed treatment. There has been research conducted in Canada and neighboring states of Montana looking at alternative control strategies for Aphanomyces root rot. And what has been found is that apparently an increase in calcium ions in the soil can prevent these zoospores, the swimming spores of the Aphanomyces pathogen, from germinating, thereby reducing the pressure of your pulse crop being infected. So adding calcium to your soil might be a strategy to reduce Aphanomyces root rot, but of course this has to be weighed against the feasibility of applying and incorporating additional or calcium into your soil. Research has also shown that brassica cover crops may have something of a biofumigating effect when decaying in the soil, suppressing oospore germination. So this might be another strategy to further explore. But again, we have to weigh the effects from this specific management strategy against the costs of adding green manure and incorporating it into the soil and what that does to soil compaction, for example. Another interesting finding is that there seems to be evidence that oats stimulate the germination of oospores, but oats are not a host to this pathogen. So including oats in your rotation might be a way of stimulating the pathogen to grow out without finding ground to actually grow on. All this information that I just cited here, you can find if you're following the link, which I included on the slide below, which is going back to very informative research presented from Alberta, Canada. Talking about the potential effect of oats on reducing Aphanomyces disease pressure in the soil, I want to highlight one of the experiments that a PhD student, Carmen Murphy, in Dr. Burrows Research Lab is currently conducting. And overall, in her thesis, she is looking at mitigation strategies for Aphanomyces root rot in pulse crops. And the specific study that I want to mention here today is a greenhouse study where she's looking at the effect of different non-host crops included in a rotation and how that might affect the disease severity of Aphanomyces in a subsequent pea crop. So the way she approached this experiment is that she created an Aphanomyces inoculum from isolates that she obtained from fields in Montana during her field surveys. And so then she inoculated this greenhouse soil with this Aphanomyces inoculum. And in this inoculated soil, she then grew various non-host crops, various cereal crops for about 3 weeks. And after that, these crops were harvested and a pea crop was planted in the very same soil as well. That was then grown for another 3 weeks. And this pea crop is essentially like a bait crop because field peas are very susceptible to Aphanomyces. So after these 3 weeks, she would take out these plants and wash the roots and then look at the symptoms developed on the pea roots to assess disease severity in response to the preceding cereal host or a cereal crop that grew in those pots before. So what you see here are the results from one greenhouse study that she conducted. On the y-axis, you see the disease severity index, which is an expression of how intense disease symptoms expressed on the roots. On the very right here, this right column shows the pea bait plant that was planted in a pot where pea was planted before. So essentially simulating a rotation of pea on pea. And you can see that disease severity on this pea bait plant following pea was considerably higher than any of the other rotation hosts that were included in the study. Disease severity was generally low for all the cereal hosts included in the study, and kind of somewhat confirming the research that I presented in 2 slides ago, shows here that really disease severity on pea roots that followed oat, there is virtually absent. So proving or showing some evidence that there might be an effect of oat to reducing disease pressure in the field. Now, I want to caution with this study that this has been conducted in a greenhouse. It has only, this specific slide only shows the results of one study. So further replications in the greenhouse as well as in the field are necessary to confirm this. But I find this personally a promising start, and that's why I wanted to share this information with you. I now want to switch to another very prevalent root rot pathogen, which is Fusarium, causing Fusarium Root Rot. And I want to discuss a couple of the symptoms that are associated with this disease. This pathogen too causes a discoloration of the roots, but the discoloration is rather reddish to brown or chocolate brown to black, as opposed to caramel brown or honey brown. And this discoloration also spreads up and down the stem further into the root system, moving from the seed attachment site downward. So the symptoms sound somewhat similar. So I included this slide here to point out some differences that can, that distinct Aphanomyces from Fusarium Root Rot. So on the left, we see diseased plants that have Aphanomyces root rot. And one thing that you can observe here is that the lower leaves particularly are yellow and pale, whereas in Fusarium root rot, the above ground parts of the plant, specifically the foliage, does typically not show any symptoms that something could be wrong with the pea plants. Another thing about Aphanomyces root rot is that the symptoms abruptly stop at the soil line, so you will not see these this discoloration of the stem and root moving past the soil line, whereas with Fusarium root rot that might be possible. And additionally, as I mentioned before, the coloration of Aphanomyces root rot is rather caramel or honey brown, however you prefer to describe this color, whereas the coloration of the taproot for Fusarium root rot is rather dark or chocolate brown, but the lateral roots appear more healthy looking or just not as discolored as with Aphanomyces root rot. Fusarium root rot prefers or requires less soil moisture compared to Aphanomyces. And it typically is a disease where symptoms are expressed a little later in the season. There are several Fusarium species that can cause Fusarium root rot. And because there are several species, Fusarium root rot typically also has a broad host range. So it's not only confined to field peas or pulse crops, but it can also move to cereals, etc., etc. So that is something to keep in mind. This image here on the right is taken from a publication by Chickpea et al. And it shows a study looking at the root rot severity of different Fusarium species and also different isolates on field pea. And those isolates were collected from North Dakota, and the study was conducted, in the greenhouse, but by North Dakota researchers as well. And so in North Dakota, what the study showed is that Fusarium avineum appears to be a rather aggressive pathogen causing more severe disease symptoms compared to some of the other Fusarium species that were isolated from root rot diseased field peas. Fusarium culmorum and graminearum also seem to be species that cause rather high disease severity. Now, such a study has not yet been conducted in Montana, but I'm going to talk in just a few slides about some research efforts that are currently being undertaken here at MSU. So what is the actual impact of Fusarium root rot on your pulse crop, in this specific case on your field pea crop? So for one, Fusarium root rot can decrease emergence of field peas, therefore reducing overall stand establishment in your field. And so this specific impact will vary between Fusarium species and also location. And then secondly, really what matters is the yield, right? So Fusarium Root Rot does ultimately also decrease or reduce the yield of your pulse crops, which is why it is important to be aware of this pathogen and do the best you can to mitigate the impact of this disease. As promised, I want to briefly mention some research efforts that are being undertaken here at Montana State University. So the previous slides all more or less refer to field peas because a lot of the research on Aphanomyces Root Rot, but also Fusarium Root Rot was done on field peas. But a recent research initiative at MSU, North Dakota State University, and other states in the Pacific Northwest and Central North of the United States is actually focusing on the Fusarium Root Rot complex in lentils. And here on the slide, you can see two students in Dr. Mary Burrows' lab that are working on parts of this project. The overall goals of this project, which has been ongoing for 2 years now and will be going on for another 2 years, is to describe the major species causing Fusarium Root Rot in lentil and explore interactions between those species. Also understand what the role of seed transmission in Fusarium Root Rot disease development in lentils is, and to look at the role of agronomic practices in establishing healthy crops and managing disease, etc. So this research is really designed to better understand this disease complex here in Montana and provide growers with helpful information on how to mitigate this disease. So to summarize this root rot section, I want to point out some more or less general management recommendations, which apply not only to Fusarium root rot, but also Aphanomyces root rot and other root rot pathogens. So generally, achieving a good stand establishment and avoiding stress and injury to your crop are very important things to pursue to set up your crop for best possible success. When it comes to soil conditions, excuse me, in general, because many of the root rot pathogens prefer high moisture in the soil, specifically oomycetes, good drainage of your soil can be very helpful in reducing the risk of these root rot diseases occurring. Avoiding or implementing strategies that avoid compaction of the soil is another important management option to keep in mind, and really spreading out the pulse crop rotation or the frequency of pulses in your crop rotation in the field can be very helpful in delaying the buildup of pathogen and inoculum in your soil. So as mentioned before, 6 to 8 years or four years at a minimum of rotating out of pulses would be ideal. 6 is better, 8 is even better. You have to kind of weigh the, between what is feasible for you and what is recommended from, our little ivory tower here. On the seed management side of things, what is really important is to plant high quality seed and apply seed treatments to suppress infection of your seed and seedlings early in the growing season. So which seed treatment is best for your particular crop and field will depend on what is the predominant root rot pathogen that you may have detected in your field. Generally, we recommend seed treatments that are a mixed mode of action, so have various mode of actions included. That doesn't only apply, for, as I mentioned before, Aphanomyces are controlled by ethaboxam, but that doesn't control Fusarium, for example. But obviously there are other pathogens that are transmitted on the seed, which may not cause root rots, but other diseases of your pulse crop that you want to try and suppress as well. I'm going to mention in a little bit a link to a seed treatment guide provided by Montana State University, where you can find more specific information. In the meantime, I also want to point out resources made available by the North Dakota State University. Following this link here, they have a very nice handout describing root rot management strategies for lentil and dry pea. And also I want to point back to this video that I mentioned initially, which has some good pointers to keep in mind as well. All right, I want to now move on from persistent below ground pathogens to a persistent pathogen that you can observe on the above ground parts of the plant, and that is the infamous Ascochyta blight pathogen. I'm sure that you're all more or less familiar with this pathogen, so I'm going to use these slides to refresh your memory and point out some important information as we're moving into the next growing season, which will hopefully be a good one. So Ascochyta blight is another disease or pathogen complex, meaning that it is composed of different species that are all causing this the same disease, but on different crops. So field peas know at least four pathogens that can cause Ascochyta blight. They have all many names because this specific genus frequently undergoes phylogenetic reorganization. So don't be discouraged. Ascochyta pisci, Didymella pisci, are almost like synonymous names. The good news is that for both lentil and chickpea, there's only at this point just one pathogen known to infect the specific crop. And another thing to keep in mind is that these Ascochyta pathogens are host specific. So those pathogens that infect pea do not infect chickpea and vice versa. So a brief review of some common telltale symptoms and signs of Ascochyta blight on pea, lentil, and chickpea. So typically you will observe these lesions on the foliage, but they can also occur on the stems, the flowers, the seed or the pods. And often these lesions are first brown, tan brown, and then you will observe these concentric rings emerging on those lesions, which you can see here and there. And if you had a hand lens, a magnifying glass, or maybe even a microscope and had a little closer look at those concentric rings, you may observe some these fungal structures, which we call pycnidia, which are structures that hold asexual spores of the pathogen, which cause reinfection of or infection of adjacent crops throughout the season, perpetuating the disease. On the field level, you will observe the discoloration or the yellowing and browning of the foliage of your crop. And if the disease progresses and the symptoms become severe, you will observe defoliation of your crop, which gives this disease the name Ascochyta blight. I want to briefly discuss the disease cycle of Ascochyta blight, in this case specifically for chickpea, but the disease cycle is essentially very similar between chickpea, lentils, and field peas as well. So this pathogen enters the field either through seed, it can be seed transmitted, or it can be introduced in the field by wind-dispersed ascospores, which are the sexual spores of this pathogen. Or the pathogen may already be present in the field and it survives on infested residue between crops. Ascospores typically develop at cool temperatures, so over the winter, whereas the infection itself and the progression of the disease is rather promoted by moderately warm temperatures ranging between 18 and 29 degrees Celsius. So the disease really starts to pick up and spread in your field in the, let's say, early spring and summer months. What is particularly challenging about this disease is that it is polycyclic. Polycyclic meaning that this disease can have several, or the pathogen can undergo several phases of infection, disease expression, spore formation, and spore dispersal throughout the season. And how often or how many cycles will occur throughout the season depends predominantly on environmental conditions. So a season that is characterized by frequent rain events and high humidity will promote the disease quite effectively. The spores, both ascospores, but also those asexual conidia spores, are spread between plants and between fields, or within the field, they are spread through water splashes. So that's why rain events really can promote the disease. Or if water drops carrying those spores are blown by the wind to adjacent fields, this can also spread the disease beyond your immediate field. And what makes this disease additionally challenging is that it can virtually occur at any time during plant development. So as I mentioned before, seedlings can be infected, foliage can be affected, stems, the flowers, the petals, the pods, as well as the seed itself. So really there is no, time out for the crop once we have this pathogen in the field and conditions are favorable for disease development. I mentioned all these points here already, so I'm just going to focus on these last 2 bullet points that refer to, challenges when it comes to managing Ascochyta blight. So dense crop canopies really promote high humidity in the underneath, and high humidity is something that favors spore production and infection of your crop with this pathogen, and also makes fungicide applications difficult. So this is another complicating factor for mitigating or managing Ascochyta blight. And the reason why we need to keep talking about Ascochyta blight too is that it has a high risk of developing fungicide resistance. In fact, strains that are resistant to strobilurin fungicides have already been identified here in Montana, which essentially means that we cannot use these strobilurin fungicides to control Ascochyta blight anymore. And that limits our options for fungicides that can be used, which I'll be talking about in just a few slides, so bear with me. So because fungicide resistance development is of concern, especially for Ascochyta blight and chickpea, we need to make sure that our management approach for this disease incorporates more than just the chemical strategy of fungicides. And so I want to point out here some other management strategies that can help reduce initial inoculum and disease pressure in your field. If you have a history of Ascochyta blight in your field, I generally recommend to plant resistant varieties. At this point, I'm talking specifically about Ascochyta blight and chickpea. So the varieties that have some resistance against Ascochyta blight are CDC Frontier and CDC Orion. If you are interested in pea or lentil varieties that are resistant against Ascochyta blight, please ask me after this talk. I mean, ask me by email, because I can tell you I do not know them from the top of my head, but I can do some research to find out some varieties that might be suitable for these other pulse crops. A very important key with Ascochyta blight management is to start with healthy, disease-free seed and apply a good seed treatment to suppress the spores that might potentially be on your seed. And I'm emphasizing this because research has shown that transmission of Ascochyta from seed to seedling can be as high as 15%. So if you have this disease on your seed, chances are that it will actually carry over to your seedling, getting an early start on your crop in the season. And the other issue here is that this pathogen really has a high epidemic potential. So an infection of less than 1% of seedlings can really, under favorable environmental conditions, lead to a serious epidemic in your field, potentially devastating your crop. Fortunately, there is help available here at Montana State University, and I want to pitch or make a little pitch for the Regional Pulse Crop Diagnostic Lab, which is dedicated to testing your seed lots for various seed-transmitted pathogens to ensure that you have a healthy seed lot to start into the season with. This slide here shows a summary of the seed lot testing that has been conducted in 2020 up until early November. And so what you can see here is how many seed lots of a total of 175, 121, and 69 of chickpea, pea, and lentil respectively were tested positive for Ascochyta, botrytis, and some other seed-transmitted pathogens. And so what I want to bring across here with the slide is that Ascochyta is really the most frequent pathogen detected on these pulse crop seed lots. And it's also worth mentioning that there are thresholds for Ascochyta as well as some other seed-transmitted pathogens as to how much incidence in a seed lot is acceptable and at what point we generally do not recommend using that particular seed for planting. And so it just so happens that there's essentially a no tolerance threshold for chickpea. So 0% incidence of Ascochyta on chickpea seeds is recommended, whereas that threshold is slightly higher. So this means that essentially all those 24 seed lots that were tested positive for chickpea should have been discarded and not used for seed in the next season. As I mentioned before, seed treatment is another important control strategy. And so this is a fungicide and disease efficacy table that we put out every year or update every year, which is available at the Montana State University website. You can follow either of those two links to get to this table. And so what I want to point out here is that looking at mixed mode of actions for Ascochyta to control Ascochyta, but also other seed and seedling pathogens, there is a whole host of fungicide options, fungicide seed treatment options available for you to really suppress an early start of Ascochyta disease on your crop. Some other pointers when it comes to Ascochyta management is that you want to, again, go back to a good rotation. You do not want to plant chickpea in a field that has been planted to chickpea in the previous years. And that is because this pathogen can survive on crop residue for several years or for up to 2 years. So you kind of want to rotate out of chickpeas for at least 2 years. Another thing that can be very effective is reducing crop density where positive to really decrease that general humidity underneath the canopy that promotes infection and disease development. And because this disease has such an explosive potential to quickly spread from just a few infected plants to a whole field suffering from this disease, you really want to closely and regularly scout so that you're able to apply fungicides upon first detection of Ascochyta blight symptoms on your crop. These two links, which I think are coming up on the next slide as well, are just links to some resources where you can find helpful information on specifically foliar fungicide applications or foliar fungicides that are very effective in controlling Ascochyta blight. I tried to create a very, very brief summary of what fungicide management programs you may implement, but I really encourage you to check out specifically the North Dakota State University website of Dr. Michael Wunsch, who is very active in exploring the efficacy of very different fungicides in controlling Ascochyta blight. So as I mentioned before, Ascochyta blight of chickpea has developed a resistance to strobilurin. So this group of fungicides is generally not recommended to be used for controlling this disease, kind of limiting the options that we have available now for this disease management. Typically, if symptoms develop early on and the canopy is not yet closed, an application of chlorophyllonyl, which is a contact fungicide, can be very effective. There are several products which include this active ingredient, namely Bravo Weatherstick and Echo 720. Because this is a contact fungicide, it is very important that you achieve good coverage, which is why this is something that is suitable only before the canopy closes. After the canopy has closed, you may have to apply fungicides in intervals of 10 to 14 days, which predominantly depends on rainfall patterns, as rainfall is what promotes the sporulation and spread of Ascochyta spores. But rainfall is also what washes off the active ingredient chlorophyllonyl from the canopy, because it is a contact contact fungicide that does not actually penetrate the plant tissue. So after canopy closure, a couple of other mode of actions are available to you, namely triazoles and carboxamides. Now, not each active ingredient that falls in the triazole or carboxamide mode of action group is equally effective. And this is where I want to direct you to this website of Dr. Michael Wunsch, which is shown here below, because he really lines out the different efficacies of different products. So in his studies, he found that Proline really is very effective in controlling Ascochyta blight, even more so than Quash or Quilt. However, I put a little asterisk here because I think it was 2 years ago, there were some observations that potentially indicate a resistance that has developed in Ascochyta to Proline, which is currently under investigation. So we may or may not be able to continually use the specific product. Generally, I recommend using mixed mode of actions. And so research also by Dr. Michael Wunsch has shown that combining these DMI or SDHA fungicides with the contact fungicide chlorophyllonyl or Bravo weather stick can really increase efficacy in controlling this disease. Overall, I really want to point out it's important that because with this disease, you will likely need to apply several fungicides several times throughout the season. It's very important to rotate between chemistries, so to reduce the risk of this pathogen developing resistance to either of those chemistries due to repeated application. I want to take the last, well, I guess I should stop here and see if there are any questions. As usual, I'm talking way longer than I should. I do not know if I can see the questions or if you're going to read them to me. If there are none, I'll be happy to keep on talking, but I guess I should check in so we're not running over time too much.
As of right now, Dr. McKelvy, there are no questions in the chat box.
All right.
We just had one pop in here, so.
Okay.
Somebody had heard that farmers were intercropping chickpeas and flax with some evidence that suggesting that it controlled Ascochyta. Has there been any research done on this?
Yeah, so hi, Andy. So I know that there has been research done on that, certainly in Canada, and I believe to know that actually Frankie Crutcher up at the Sydney Research Center is currently doing some studies looking at that with a master's student. So specifically from Montana, I don't think we have yet a whole lot of, research information available since that is an early study, but there are certainly efforts being undertaken to kind of look at the potential benefits of intercropping. I myself have talked to growers as well in northern Montana who have reported that that was an effective, or that strategy worked for them in reducing the spread and impact of Ascochyta blight. And the reason why that probably works is that flax is not a crop, not a host to Ascochyta blight. And so just having, virtually like a barrier of a non-host crop between susceptible plants just really reduces the spread of these pathogen spores between rows and therefore reduces the spread of the disease throughout the field. One thing to keep in mind in that is that apparently you want to orient your rows perpendicular to the wind direction. So you want to make sure that the way the wind blows, the intercropping rows actually are catching the spores. So you don't want the wind blowing through the rows, but actually being blocked by the rows. I hope that was helpful. Cool. Well, how does it look in time? I guess we're out of time. So I guess I should stop here. If that's the case, then I want to thank everyone for attending and for listening. And I really want to encourage everyone to, reach out to myself or consult the resources that I've shown initially and shown here again too for further information or with any questions that you might have. We're here to help and we're happy to do so.
All righty. Well, thank you again, Dr. McKelvy, for joining us. That was super, super informative. So thank you and the team at MSU for bringing us this information.
My pleasure.
Thanks again, everybody, for joining us for the Great Montana Ag Rally. We really hope you found this informational. Thanks to our speaker, Dr. Uta McKelvy, and BASF for sponsoring this session. I thought this was a great session. Thanks again for attending, and Happy New Year to everyone.